Welcome back. Now we discuss about the third step of rDNA technology. In previous video we discussed about the two step of rDNA technology, that is selection and isolation of desired gene and selection and isolation of plasmid, uh, nothing but a vector, selection of vector. So we vector selected as a plasmid and desired gene is the human growth hormone gene or gene for human growth hormone, or we can take the insulin gene from the beta cells of pancreas. Now this desired gene and this plasmid, so first of all this plasmid is cut by the restriction enzyme, and then this desired gene and this plasmid is joined to get this rDNA recombinant DNA molecule. Okay, this desired gene is inserted into the marker gene. It inserted into the marker gene, or is also called is marker gene, or for example the antibiotic resistance gene like tetracycline or streptomycin resistance gene. So this DNA is inserted into that, and then after the formation of recombinant DNA, this recombinant DNA is inserted into this bacteria or any host, prokaryotic or maybe eukaryotic, maybe in a yeast cell or in a bacterial cell. And after the insertion of recombinant DNA into the bacteria, then these bacteria start to multiply, and along with the multiplication these our desired genes also multiply. Okay, so this is all about the insertion of desired gene into the vector by using the restriction enzyme, and this whole process is called a gene splicing. Now next step, so in the next step is introduction of this recombinant DNA into the suitable host cell and multiplication of gene. So now that recombinant DNA that made, that recombinant DNA is inserted into the bacteria or any host cell. How that inoculate, how that inserted, that we see. So first, introduction of recombinant DNA into the suitable cell and multiplication of genes. So here we introduce a recombinant DNA, that is now we made that in a suitable host cell, maybe the is bacterial cell or maybe yeast cell, mean prokaryotic cell or in a eukaryotic cell. Then this recombinant DNA contain or have the our desired gene, and that desired gene is multiply in that bacterial cell when that bacteria is start cell division. Okay, next is, so in this there are number of process. So for the introduction of desired gene, of introduction of recombinant DNA into the suitable vector, we have the different techniques. For example, gene transfer technique. So for the gene transfer, depending upon the target cell and objective of gene manipulation, so objective of gene manipulation and target cell, on that the gene transfers depend. So there are two methods mainly for the introduction of this recombinant DNA into the or desired gene into the host cell. Two method, number one is a natural method, number two is a artificial method. So for the natural method, so in a natural method there are number of in a natural method there is a number of methods. So number one, the conjugation by the bacterial transformation, transposition, phase transduction, retroviral transduction, and the vector mediated transfer. So this is the technique. So first we see the list of technique and then we discuss one by one. Next is the artificial technique. So in artificial technique, number one, a physical method. So in artificial method there is a two method, physical and chemical. So in a physical method, for example, micro injection, three method, physical, chemical and electrical method. So first in a physical is a micro injection, this by micro injection, then by bioelectric, that is micro projectile gun, and last by the protoplasmic fusion. Next in a chemical method for the of gene transfer, we require calcium phosphate mediated gene transfer. Next, use of polyethylene glycol PEG. Next, use of DEAT DEAE dextrum. Next, use of liposome. Next, so in the next video we discuss about in one by one these techniques. Thank you. I hope this video is helpful for you.